I've talked about some of these projects before, about the human genome and what that might mean, and discovering new sets of genes. We're actually starting at a new point. We've been digitizing biology, and now we're trying to go from that digital code into a new phase of biology with designing and synthesizing life. So we've always been trying to ask big questions. What is life is something that I think many biologists have been trying to understand at various levels. We've tried various approaches, paring it down to minimal components. We've been digitizing it now for almost 20 years when we sequenced the human genome. It was going from the analog world of biology into the digital world of the computer. Now we're trying to ask, can we regenerate life? Or can we create new life out of this digital universe? This is the map of a small organism, Mycoplasma genitalium, that has the smallest genome for a species that can self-replicate in the laboratory. And we've been trying to just see if we can come up with an even smaller genome. We were able to knock out on the order of 100 genes out of the 500 or so that are here. But when we look at its metabolic map, it's relatively simple compared to ours. Trust me, this is simple. But when we look at all the genes that we can knock out one at a time, it's very unlikely that this would yield a living cell. So we decided the only way forward was to actually synthesize this chromosome so we could vary the components to ask some of these most fundamental questions. And so we started down the road of, can we synthesize a chromosome? Can chemistry permit making these really large molecules where we've never been before? And if we do, can we boot up a chromosome? A chromosome, by the way, is just a piece of inert chemical material. So our pace of digitizing life has been increasing at an exponential pace. Our ability to write the genetic code has been moving pretty slowly, but has been increasing. And our latest point would put it on now an exponential curve. We started this over 15 years ago. It took several stages, in fact, starting with a bioethical review before we did the first experiments. But it turns out synthesizing DNA is very difficult. There's tens of thousands of machines around the world that make small pieces of DNA, 30 to 50 letters in length. And it's a degenerate process, so the longer you make the piece, the more errors there are. So we had to create a new method for putting these little pieces together and correct all the errors. And this was our first attempt, starting with the digital information of the genome of Phi-X174. It's a small virus that kills bacteria. We designed the pieces, went through our error correction, and had a DNA molecule of about 5,000 letters. The exciting phase came when we took this piece of inert chemical and put it in the bacteria. And the bacteria started to read this genetic code, made the viral particles. The viral particles then were released from the cells and came back and killed the E. coli. I was talking to the oil industry recently, and I said they clearly understood that model. They laugh more than you guys are. And so we think this is a situation where the software can actually build its own hardware in a biological system. But we wanted to go much larger. We wanted to build the entire bacterial chromosome. It's over 580,000 letters of genetic code. So we thought we'd build them in cassettes the size of the viruses, so we could actually vary the cassettes to understand what the actual components of a living cell are. Design is critical, and if you're starting with digital information in the computer, that digital information has to be really accurate. When we first sequenced this genome in 1995, the standard of accuracy was 1 error per 10,000 base pairs. We actually found on resequencing it 30 errors. Had we used that original sequence, it never would have been able to be booted up. Part of the design is designing pieces that are 50 letters long that have to overlap with all the other 50-letter pieces to build smaller subunits. We have to design so they can go together. We design unique elements into this. You may have read that we put watermarks in. Think of this. We have a 4-letter genetic code, A, C, G, and T. Triplets of those letters code for roughly 20 amino acids, that there's a single letter designation for each of the amino acids. So we can use the genetic code to write out words, sentences, thoughts. Initially, all we did was autograph it. Some people were disappointed there was not poetry. We designed these pieces so we can just chew back with enzymes. There's enzymes that repair them and put them together. And we started making pieces, starting with pieces that were 5,000 to 7,000 letters, put those together to make 24,000 letter pieces, then put sets of those going up to 72,000. At each stage, we grew up these pieces in abundance so we could sequence them, because we're trying to create a process that's extremely robust that you can see in a minute. We're trying to get to the point of automation. So this looks like a basketball playoff. When we get into these really large pieces, over 100,000 base pairs, they won't any longer grow readily in E. coli. It exhausts all the modern tools of molecular biology, and so we turned to other mechanisms. We knew there's a mechanism called homologous recombination that biology uses to repair DNA that can put pieces together. Here's an example of it. There's an organism called Deinococcus radiodurans that can take 3 million rads of radiation. You can see in the top panel, its chromosome just gets blown apart. 12 to 24 hours later, it put it back together exactly as it was before. We have thousands of organisms that can do this. These organisms can be totally desiccated. They can live in a vacuum. I am absolutely certain that life can exist in outer space, move around, find a new aqueous environment. In fact, NASA has shown a lot of this is out there. Here's an actual micrograph of the molecule we built using these processes, actually just using yeast mechanisms with the right design of the pieces. We put them in. Yeast puts them together automatically. This is not an electron micrograph. This is just a regular photomicrograph. It's such a large molecule, we can see it with a light microscope. These are pictures over about a 6-second period. So this is the publication we had just a short while ago. This is over 580,000 letters of genetic code. It's the largest molecule ever made by humans of a defined structure. It's over 300 million molecular weight. If we print it out at a 10 font with no spacing, it takes 142 pages just to print this genetic code. Well, how do we boot up a chromosome? How do we activate this? Obviously, with a virus, it's pretty simple. It's much more complicated dealing with bacteria. It's also simpler when you go into eukaryotes like our cells. You can just pop out the nucleus and pop in another one, and that's what you've all heard about with cloning. With bacteria and archaea, the chromosome is integrated into the cell. But we recently showed that we can do a complete transplant of a chromosome from one cell to another and activate it. We purified a chromosome from one microbial species. Roughly, these two are as distant as human and mice. We added a few extra genes so we could select for this chromosome. We digested it with enzymes to kill all the proteins. And it was pretty stunning when we put this in the cell, and you'll appreciate our very sophisticated graphics here. The new chromosome went into the cell. In fact, we thought this might be as far as it went, but we tried to design the process a little bit further. This is a major mechanism of evolution right here. We find all kinds of species that have taken up a second chromosome or a third one from somewhere, adding thousands of new traits in a second to that species. So people who think of evolution as just one gene changing at a time have missed much of biology. There's enzymes called restriction enzymes that actually digest DNA. The chromosome that was in the cell doesn't have one. The chromosome we put in does. It got expressed and it recognized the other chromosome as foreign material, chewed it up, and so we ended up just with the cell with the new chromosome. It turned blue because of the genes we put in it. And with a very short period of time, all the characteristics of one species were lost, and it converted totally into the new species based on the new software that we put in the cell. All the proteins changed, the membranes changed. When we read the genetic code, it's exactly what we had transferred in. So this may sound like genomic alchemy, but we can, by moving the software of DNA around, change things quite dramatically. Now, I've argued this is not Genesis. This is building on 3.5 billion years of evolution. And I've argued that we're about to perhaps create a new version of the Cambrian explosion where there's massive new speciation based on this digital design. Why do this? I think this is pretty obvious in terms of some of the needs. We're about to go from 6.5 to 9 billion people over the next 40 years. To put it in context for myself, I was born in 1946. There's now 3 people on the planet for every one of us that existed in 1946. Within 40 years, there'll be 4. We have trouble feeding, providing fresh, clean water, medicines, fuel for the 6.5 billion. It's going to be stretched to do it for 9. We use over 5 billion tons of coal, 30 billion plus barrels of oil. That's 100 million barrels a day. When we try to think of biological processes or any process to replace that, it's going to be a huge challenge. Then, of course, there's all that CO2 from this material that ends up in the atmosphere. We now, from our discovery around the world, have a database with about 20 million genes. And I like to think of these as the design components of the future. The electronics industry only had a dozen or so components, and look at the diversity that came out of that. We're limited here primarily by a biological reality and our imagination. We now have techniques, because of these rapid methods of synthesis, to do what we're calling combinatorial genomics. We have the ability now to build a large robot that can make 1 million chromosomes a day. When you think of processing these 20 million different genes or trying to optimize processes to produce octane or to produce pharmaceuticals, new vaccines, we can change just with a small team, do more molecular biology than the last 20 years of all science. And it's just standard selection. We can select for viability, chemical or fuel production, vaccine production, et cetera. This is a screen snapshot of some true design software that we're working on to actually be able to sit down and design species in the computer. We don't know necessarily what they look like. We know exactly what their genetic code looks like. We're focusing on now 4th generation fuels. You've seen recently corn to ethanol is just a bad experiment. We have 2nd and 3rd generation fuels that will be coming out relatively soon that are sugar to much higher value fuels like octane or different types of butanol. But the only way we think that biology can have a major impact without further increasing the cost of food and limiting its availability is if we start with CO2 as its feedstock. And so we're working with designing cells to go down this road, and we think we'll have the first 4th generation fuels in about 18 months. Sunlight and CO2 is one method. But in our discovery around the world, we have all kinds of other methods. This is an organism we described in 1996. It lives in the deep ocean, about 1.5 miles deep, almost at boiling water temperatures. It takes CO2 to methane using molecular hydrogen as its energy source. We're looking to see if we can take captured CO2, which can easily be piped to sites, convert that CO2 back into fuel to drive this process. So in a short period of time, we think that we might be able to increase what the basic question is of what is life. We're truly, have modest goals of replacing the whole petrochemical industry. If you can't do that at TED, where can you? Become a major source of energy. But also, we're now working on using these same tools to come up with instant sets of vaccines. You've seen this year with flu, we're always a year behind and $1 short when it comes to the right vaccine. I think that can be changed by building combinatorial vaccines in advance. Here's what the future may begin to look like with changing now the evolutionary tree, speeding up evolution with synthetic bacteria, archaea, and eventually eukaryotes. We're a ways away from improving people. Our goal is just to make sure that we have a chance to survive long enough to maybe do that. Thank you very much.
Unbelievable. Unbelievable. We're going to sit here. Come and sit this way. Craig, you have an incredible way of just saying in a very calm way stuff that will probably change not our lives, but for our children in the world to come. It's an unbelievable talk. Help me understand a couple of things, and we're going to have some questions from the audience in a bit, so be ready in a few minutes to put a hand up. Short, crisp questions, less than 30 seconds. You've discovered all these millions of genes and organisms in your ocean voyage, all of which do things that really we weren't aware of. Why not use some of those organisms rather than designing from scratch? Is that whole process really necessary to achieve these incredible goals you have?
It's an important question. I mean, right now we are finding a lot of biodiversity. We find organisms in the environment that actually produce octane. You grow these in the lab, they smell like a gas station or a diesel tank, but nothing ever evolved to produce the kind of quantities we need. And the scale is what's really important. So we were thinking of making micro-refineries. So if you make a micro-refinery that makes 20,000 liters a day, you need 1 million of those to match what we use in oil. So the scale is what's critical. And it's, while metabolism can change by 1,000 to 1 million fold in the lab, it's not clear anything can really work on the scale that we need. We and others are trying to change existing organisms now. That's a stopgap to get going. The ultimate will be chemical design. We're talking to most of the chemical industry that use oil and coal as raw materials to see if it can switch capturing CO2 back in these processes. So I think we eventually have to get to biodesign and to optimize these. We absolutely have to do that.
So there's no reason why nature itself would have produced bugs with exactly the properties that we need right now?
In fact, that's why photosynthesis is so ineffective. In fact, if things produce things on the scale we need, this would be a methane planet, for example, not a oxygen-driven one.
Huh. And so you've got this three-part process for creating synthetic life. Two of the parts are completed. The first part was you basically proved that you could move a natural chromosome from one bug to another and reboot that bug.
And we've made now the chemical molecule, and they're making even larger ones. So we are now booting up the chemically made chromosome. And that would I think that's a technical difference, but that'll be the third absolute proof of the whole process.
So right now in your lab, scientists are trying to take that huge chromosome you referred to, insert it into
another
bacteria, and hope that it boots and basically starts reproducing using that.
We try not to base too much on hope, but.
But how is it? Are you waiting? I mean, do you have your cell phone switched on, waiting for the call? Is this surely this is a big deal for you
Yes. They're very slow experiments. It takes about 6 weeks or so because these cells, being so small and so limited, grow very slowly. But I think it will be exciting when that is booted up, and I'm sure it will happen this year. But there's a lot of barriers to doing this. The combinations have to be right. You have to have the right restriction enzymes. Cells, you can see maybe why they would have evolved to not want this to happen. If you can lose your entire identity just from having DNA exchange, it would change the nature of sex quite a bit. So when that's happened,
So when that's happened, you then have this ability to, you've got a chromosome that you can then add different design elements onto, try out different life forms. Talk to us more about that process and how it is right now. I mean, right now, is it possible on a computer to say, we think that this particular chromosome will generate a bug that does X?
Yeah, we're absolutely using software to design the pathways, the metabolic organisms, all the aspects we want in the cells, and we're building those pathways. So it is absolutely based on computer design. Even trying to change existing cells, you have to put in whole new chemical pathways. DuPont spent 10 years and about $100 million changing about 16 different genes in E. coli, where they go in massive tanks going from sugar to propanediol. We're trying to not just do it by trial and error, we're trying to do it by direct design. And I think it will change the process, change the efficiency, and get us there much faster. But think of this as an iterative process. You know, a lot of people like to think in terms of genesis, or we're creating life from scratch. We're using one of the key tenets of life. All life derives from other life. We're using that 3.5 billion years of evolution. We're just trying to take it over now with new software to take it very rapidly in new directions. So the first cell, now you can design it so it can easily take new chromosomes, new material to get to the next stages. So we'll see an exponential change in the pace of the sophistication of the organisms and what they can do.
So just building on that, I mean, you had that interesting slide up saying software that creates its own hardware.
Yeah.
Just talk about that a bit more, because someone might say, this isn't really new synthetic life. All you're doing is you're writing code. You're making a computer disk. You're not making a computer.
We're definitely not creating life from scratch. It's an intellectual concept. We absolutely start with digital code in the computer, make that chemical molecule. We start with 4 bottles of chemicals, but we are absolutely dependent on having life evolve to this point to be able to take that software and read it. So it's like if you had a piece of software and you had no computer to read it on, it doesn't do you any good. But we can use those basic tools to get very rapidly to whole new orders of magnitude.
I mean, I understand you to say that you actually don't need to make all of life from scratch. The key thing is just to make the code, because that then does the rest of the job for you.
Exactly.
Yeah, so that's really fun. And are you using, it seems like part of the power of this is that you don't have to like design one chromosome and then do it. Part of what you're doing is creating many designs at once and throwing them out there and seeing which ones work in a controlled environment?
Yeah, because the vastness of biology, right now with a 20 million gene data set, at the pace of science, if 10,000 of those get understood in a decade, with the way science is going to be really stunning. So we have to have these combinatorial approaches. We have to have the ability to do basically millions of experiments simultaneously using empiricism. But it's all the question you asked. If you just want to see what leads to a life form growing in these conditions, that's one question. But if you're screening for new derivatives of octane to be efficiently produced, starting with CO2, and you want to know that, you can optimize for that question as well. So like all biology, it's based on selection, and it's based either on the natural environment that you're selecting for or the constraints and questions you put on it.
So you're kind of creating your own Darwinian environment, throwing in lots of thousands, millions of competing designs, and seeing which superbug comes out of it that, whoa, this thing actually gobbles up CO2 and makes energy. I mean, that's going to be a big day for mankind, isn't it, when you find that one?
We are doing that right now this year with pathways. So we're working with a eukaryotic system where we just make one artificial chromosome with these synthesis pathways. Once you know the 50 genes that you want in, we have for each of those 50 genes 10 to 100,000 different varieties. So we can substitute those in and try and optimize the system. So biology is empirical at this stage because there is so much diversity. The combinatorial, computational aspects allow us to overcome that diversity.
Fantastic. Question from the audience. Chris, questions? I saw this hand first.
I can make the case that you and your company are the most dangerous humans on planet Earth, because just as you have opening up the door to wonderful aspects, weaponization through the same process could do in the human species.
Was that the question?
That's the question. What do you guys do for security?
It is an important question, one that's been raised from the beginning, from the first bioethical review that suggested that we should go ahead with the caveat. Fortunately, there's not that many people on our planet wanting to do harm with these tools. Part of a review funded by the Sloan Foundation with us and MIT looked at this question and, along with the government, decided very few biological agents that we work with or even could be weaponized would people go to this route. There's 10,000 freezers around the world with hoof and mouth disease virus in it that you wouldn't go to try and build the sophistication to do it. But it is an important issue. Every new technology has the ability to be abused.
You had on your slide, you had an intriguing little label on one of the boxes that said suicide gene. Well, what is that?
Well, we build in various levels of security with roughly 10 million experiments of people putting all kinds of different genes in E. coli over the last 30 years. There's never been one issue with it because the E. coli was modified never to survive outside the lab. So we use those as starting positions, but extra security is a suicide gene that if ever got out, you could easily trigger that and it would kill the organism. So,
but if what you're building is a 21st century version of PageMaker or something, so that in the natural consequence of all this is in 30 years' time, any grad student in their lab can build up on screen, I want this, press a button, here's my super designed, self-replicating, kill the human race bug. Aren't you creating the possibility for that to happen?
Well, it's actually, fortunately, creating pathogens is probably one of the most difficult things. Out of the repertoire of probably 50 million species on our planet, it's only a few dozen that take any interest in us, and it's even hard to understand that. There's courses now at MIT and Berkeley teaching these methods, and some are advocating biohacking. I'm not advocating biohacking, but part of what we're working with various groups on is we need new tools against emerging infections. It really doesn't matter whether they're man-made or not. We now, in this last year in this country, drug-resistant Staph aureus killed more people than HIV. That's a new emerging infection. HIV was an emerging infection. SARS was an emerging infection. And I guarantee as we go from 6.5 to 9 billion people with increased poverty, we're going to have a lot of new emerging infections. So we need new antibiotics, new antivirals, and new vaccines. If we have those in abundance, it eliminates any threat of biological warfare.
Okay, so your tools can work for the defense as fast as for the attack, so to speak. We're going to have one more question. Is Larry Page, do you have your hand up? Did I see that? Yeah.
I guess I'm interested in the how efficient you think the photosynthesis or the CO2 based energy systems can be based on life.
Well, it's a good question and an important one. What we see with organisms like Methanococcus, CO2 is their source of carbon. So every bit of carbon in that cell is derived from CO2. What we see with plants is not very efficient. What we see with algae is much more efficient. So if we engineer algae using sunlight and CO2 and engineer the organisms like Methanococcus, we could actually use the combination of those to run day and night in a pretty efficient fashion, capturing CO2. The limitation, interestingly, is going to be CO2 for this process because we can't just capture it out of the air. It's not high enough concentration. But CO2 sequestration, where people have been talking about dumping it into coal beds and oil beds, instead we think we can take that stream, convert it back into fuel, changing the equation in real time. So we need high concentrations of CO2 to drive this process. So it's we need to drive the CO2 sequestration industry in parallel with this.
So finally, Craig, I just wanted to ask almost a more personal question about how you think of your own legacy in this. You must lie awake some nights just thinking, how did this happen? How was it me who was put in a position where potentially could materially alter the future of the human race, create this new explosion of life forms, whatever. This is a big deal. I mean, how
I don't
how
want
do
to
you
scare
think of it?
people. I don't think those things.
But that, but okay. So I mean,
But
when you
I think
first
about the potential of what we're trying to do. You know, we were at Davos. I got very depressed being at Davos this year because it was clear most of the business executives that were there sort of buy into the CO2 issue as a pain for them. The consensus was nothing's going to change in the next 40 years because of vested interests. It was pretty discouraging. So I worry much more whether these technologies and others that people here are developing can get out there in time to make a difference. We're playing a hell of an experiment with this planet that worries me more than any other aspect.
So when you first came to TED and I asked you, hey, couldn't you be accused of playing God? And you said, oh, we're not playing. It was a great joke, you know, or was it?
Well, we're not playing, for sure. And I think it's part of the ethical review that we had. Most major religions were brought, and it's part of most religions to actually play God. You're supposed to use your knowledge to improve humanity. So I think it depends on who's making the accusation. If it's religions, then it's a good thing. But we think this is science at its best, not religion.
Is there anything that the TED community can do to help you in your vision?
Well, I think, we have Al Gore here who's helped us all understand the importance of this timing-wise. I think the one thing that could kill this are the ideas that were just mentioned, that this is so scary we shouldn't do it. We shouldn't try and solve these problems this way. I think public acceptance, which is why I spend time talking to the public, is as important as the quality of the science. We saw that in Europe with GMOs. I joke that Europeans now want DNA-free food. We need real solutions. I think if people listen to this pessimistic view that came out of Davos, the urgency is not really there. We don't see it, and I think that's how this community can help the most.
Craig Venter, what you're doing is astonishing. Thank you so much for sharing that with us. Thank you. It's unbelievable. Thank you.